Here we go. Again, welcome everybody to tonight's Cafe Scientifique Twin Ports. Tonight we're going to learn about some of the dangers that surround us. Tonight we're going to learn about some of the toxins that inhabit our world. Tonight, we're going to hear from Dan Villeneuve, toxicologist at the U.S. Environmental Protection Agency. Dan was chosen to lead this Cafe Scientifique event this year because when I was learning from Dan about toxicology and why he got into it, one of the things he said to me really stuck with me, which was, I'm interested in biology and how it breaks. So from that perspective, I introduce to you our speaker tonight, Dr. Dan Villeneuve. Thank you all for attending.
Welcome, everyone. And let me echo Nate's thanks for attending tonight to hear about the scary science of toxicology. This presentation is really geared to those of you who don't dress up as a toxicologist every day of the year other than Halloween. And I'd like to start by saying that nothing I'm about to say represents the policies or positions of any organization I'm affiliated with. It's just me, and this is intended for your entertainment and for our discussion. It begins with a metallic taste on your tongue and a slight garlicky odor to your breath. Your mouth dries as you struggle to swallow. Muscles begin to cramp, numbness spreading to your hands and feet. Soon, reddish rashes erupt across your body, and intense thirst takes hold. Your stomach cramps with nausea as you double over to vomit, and worse. Confusion, paranoia, delusions, and intense drowsiness. Finally, seizures, coma, and the sweet release of death. This is not the interview with a vampire, nor your personal audition for Twilight or the Vampire Diaries. This is a first-hand account of an acute arsenic poisoning as described in the critical reviews of environmental science and technology. Toxicology is colloquially known as the basic science of poison. And I start with arsenic because perhaps no poison is as well known as arsenic. Arsenic was long regarded as the poison of kings and the king of poisons. Its toxic properties were known back to at least 370 BC, and first laws against poisoning date back to about 82 BC. Because of its potency, the ease with which it could be obtained, and the discreetness with which it could be administered, it was a favored poison of the ruling classes to kill off their rivals and adversaries throughout much of human history. In the Victorian era, Victorians couldn't get enough of it. It was said that nothing appealed more to the increasingly literate public of the Victorian era than lurid newspaper reports of murder trials involving poison. Development of the life insurance industry in the first half of the 19th century, interesting enough, corresponded to a rise in poisoning cases. And in France, arsenic actually became known as inheritance powder. Again, because it's ease to obtain, ease to administer, and difficulty proving the cause of the death. That was until around 1832, when a chemist named James Marsh came up with a method to pass hydrogen sulfide through the arsenic-laced compound to produce a yellow precipitate of arsenic sulfate that could be arsenic sulfide that could be used to detect the presence of arsenic. In 1832, police arrested a man, John Bodle, for being accused of lacing his grandfather's coffee with poison. James Marsh was brought in to provide evidence at the trial. And while James did find presence of arsenic based on his yellow precipitate, unfortunately it wasn't stable and it deteriorated by the time he got to trial. So without that forensic proof, John Bodle was acquitted and went free, later going on to admit that he had indeed poisoned his grandfather. Upset by his inability to support the conviction with his test, John Marsh dedicated himself to improving the test in subsequent years, such that by 1840, the case of Marie Lafarge, his test had improved considerably. Marie Lafarge had been seen buying arsenic to exterminate rats, she claimed, and testified that she had stirred a white powder into her husband's food. As part of the trial, that food was tested and found to be positive for arsenic using the old methods as well as the newer Marsh test, and the eggnog alone was said to contain enough poison to poison 10 persons. But when Lafarge's body was exhumed, the local chemists assigned to the case were not able to detect the poison. Looking for more definitive evidence, the defense called in Matthew Orfila, and he was able to demonstrate that the original test had been carried out incorrectly. Using new modifications to the original Marsh test, he found definite traces of arsenic in the body and showed that it did not come from the surrounding soil. Based on his evidence, Marie Lafarge was the first person convicted largely on direct forensic toxicological evidence. And Matthew Orfila went on to be recognized as one of the founders of the modern science of toxicology, notably the discipline of forensic toxicology. With an established forensic test, acute arsenic poisoning went out of style, just like my 80s haircut. Another well-known father of the field of toxicology was one Philippus Aurelius Theophrastus Bombastus von Hohenheim Paracelsus, or popularly known as just Paracelsus, who came up with the phrase that really makes up the central dogma of the science of toxicology, and that is that all substances are poison. There's none which is not a poison, and it's the right dose that differentiates the poison from remedy. With that concept in mind, many of the classic poisons that people know and think about when we say the term poison, like arsenic and cyanide, are actually middle of the pack in terms of their lethality. Cyanide, for example, was served to Rasputin in 1916 in a meal of cakes and wine laced with cyanide, but Rasputin gobbled it down with no ill effect. Again, the dose wasn't high enough to cause the toxicity, so they had to shoot him instead. It's actually nature that produces some of the most deadly poisons. Topping the scale, botulinum toxin produced by bacteria is just a protein that prevents acetylcholine release from axons, leading to paralysis and ultimately death. At about 100 times less potent are the batrachotoxins, popularly used as dart poisons in South America, derived from excretions from the skin of frogs. These steroid alkaloids prevent sodium channels from closing. Again, potent neurotoxic effects that can lead to paralysis and death. And about 100 times less potent than those, another well-known naturally produced toxin, tetrodotoxins, another steroid alkaloid neurotoxicant, popularized in the 1990s by the ethnobotanist Wade Davis, who raised the possibility that tetrodotoxins were the active ingredient in Haitian zombie powders that would induce a death-like state in the human that consumed them, leading them to be buried and then later dug up and allowed to walk among the living. With modern chemistry, humans have joined the fun, so to speak, coming up with and devising some toxic chemicals of their own. Some were made by design, so pesticides, warfare agents like VX gas, another organophosphate neurotoxicant, and some indeed were produced entirely by accident as byproducts of various industrial processes and combustion, such as 2,3,7,8 tetrachlorodibenzoparadioxin, or dioxin, which is formed when organic chemicals are combusted in the presence of chlorine and oxygen at high temperatures. These chemicals, while not directly neurotoxic, not causing the same sort of acute effects as many of these other neurotoxins, do cause a variety of chronic effects, wasting syndromes, developmental deformities, and so on that lead to a variety of adverse outcomes. And again, a highly potent compound, even though it was never intended to be produced. And in fact, as Paracelsus stated, all chemicals are toxic. And we all have, well, at least those of us who consume alcoholic beverages, have some familiarity with this concept of dose response. You know, one cocktail on a Friday evening might produce a nice stimulant buzz type of effect, but if we overdo it too much, have slurred speech, slowed thinking, loss of motor control, potentially vomiting, hopefully not unconsciousness and death that follows, but certainly too much alcohol can lead us there. We need only turn on the afternoon football game to hear all the ads for pharmaceuticals and their various side effects, and read in the newspapers about overdose related to certain drugs to understand that the dose really does make the poison. So toxicology is the scientific study of the adverse effects of chemicals and other stressors on living organisms. Spends an awful lot of time trying to characterize this particular relationship, the dose-response relationship, where concentration is depicted on the x-axis, on the y-axis, various measures of severity of biological effect, whether that be direct observation of mortality, onset of various pathologies, or various other types of outcomes. In toxicology today, acute poisoning are still the ones that make the headlines, and forensic toxicology is really the public face of the field in the popular imagination. We've all heard about deaths, unexpected deaths of celebrities, and awaiting the toxicology report to see if an overdose was potentially the cause of death. Or we see on crime shows on television forensic toxicologists that help prove the case against potential criminal. But it's environmental toxicology and kinds of chronic low dose exposures that occur every day through our interactions with our environment and products of modern life that are estimated to have the greatest global health impacts. It's estimated that pollution is the largest environmental cause of disease and premature death. Diseases associated with pollution are responsible for an estimated 9 million premature deaths in 2015, which in that year was 3 times the number of deaths from AIDS, malaria, and tuberculosis combined, and 15 times that from all wars and violence. And that's just in humans. The toll that it takes on the environment and other species is really unaccounted for. The modern face of the king of poisons, or arsenic, is really today most of that poisoning occurring through sources like contaminated drinking water, resulting in a diverse set of chronic health effects that are very difficult to differentiate from other sources of disease or dysfunction, and very hard to prove, particularly at the individual level, in terms of the exposure to the toxic chemical being the cause of the kinds of chronic effects that are observed due to the variation in lifestyle, genetic susceptibility, environment, co-occurring disease, and other factors that can shape how an individual responds to the pollutants that they're exposed to. So, is the overall message here that we just need to be afraid, very afraid of toxins all around us in our drinking water, in the foods that we eat, in the air we breathe, in the workplace, and in our homes. And really the science of toxicology and the science that we do is really aimed to avoid the need to be afraid, very afraid. And so toxicology, like all sciences, is modernizing. Scientific knowledge is expanding rapidly, and it's estimated that global scientific output doubles every 9 years. Toxicology is no different, and we've learned a lot about the myriad of ways that chemicals can harm people and harm the planet. So while toxicology has historically been somewhat of a morbid discipline that involved extensive testing of laboratory animals, where we would basically administer chemical to an animal and then observe unpleasant things happen, whether they watching animals drop dead in their cages, whether it be viewing irritation in the eyes or skin. These animals basically became our surrogates, so we didn't have to test these products on ourselves. And this kind of testing was largely used to define the concentrations that would be safe to humans and the environment, and try and assure that we kept these chemicals at concentrations that wouldn't produce adverse effects. As I said, toxicology is modernizing, and we're trying to move away from these somewhat morbid methods of the past. And so legislation like that in Europe in 2003, producing strict bans on animal testing for cosmetic products and their ingredients, have been introduced. Newer legislation implementing and promoting 3 R's principle to reduce, refine, and replace the use of animals in testing. And even more recently, new legislation and directives from the EPA administrator directing the agency to reduce the use of animal testing considerably by the year 2035. So where our toxicology of the past was really based on observing dead animals in the cage, for example, now it's really cells in a 96 well plate that have to be afraid, very afraid. We're now using more and more cell-based assays conducted in high throughput, still doing characterization of dose response and concentration response, but examining fundamental biological interactions that can lead to toxicity rather than directly observing adverse effects in animals. And so where toxicology of the past was really focused on reporting effects like mortality, pathology, deformities, and reproductive failure, things that we can all sort of wrap our heads around and understand how to interpret, now a lot of the data that we're generating produce biological activity profiles that have to be linked to other knowledge and models to really understand what those data mean in terms of their potential adverse outcomes. So with that in mind, we've taken on an effort to catalog so-called adverse outcome pathways, cataloging, if you will, the many fascinating ways to die through poisoning, and documenting how these changes, these interactions that chemicals have at the molecular and cellular level, how those changes then interact with the physiological functions of humans and other organisms to lead to the effects that we ultimately want to avoid, and trying to use these methods to be able to forecast that something bad may happen if we're exposed to too much of this chemical. Now, while we're gaining new knowledge all the time and we're becoming better and better at predicting and preventing adverse effects of chemicals, we still don't always get it right. There are surprises that occur in nature, and we base our decisions and our thinking based on the available knowledge, but idiosyncratic toxicities do occur. One example was the early 2000s, a mass die-off of oriental whiteback vultures in Pakistan, where cattle were left to lay, dead cattle were left to lay on the fields for the vultures to eat. It was found that these cattle, which had been treated with diclofenac, a common non-steroidal anti-inflammatory drug that you can obtain over the counter. It's used to inhibit cyclooxygenases, just like many other anti-inflammatories. And it's used at a therapeutic dose of about 2 milligrams per kilogram in humans, which is below its toxicity in most mammals. But unfortunately for these vultures, they're much more sensitive, largely due to some differences in their physiology. Prostaglandins, in addition to regulating inflammatory responses, also act as vasodilators in kidney, helping to control blood pressure and kidney blood flow. And because blood flow in the avian kidney is very different from that in mammals, it makes these birds much more sensitive, such that the same concentrations that are therapeutic in humans are actually 100% lethal to these oriental whiteback vultures. And so this unique confluence of a different sort of physiology, along with a cultural practice of letting these sickened cattle that were treated with this anti-inflammatory to lay in the fields and be consumed by the vultures, led to this unpredicted sort of toxic effect. There are other examples of these dramatic differences in sensitivity across species, and from all these cases, we try to learn and use what we've learned to get better at predicting and preventing these kinds of unfortunate events. And so, for example, focusing on the toxicity of dioxin in birds, there's over a 1,000-fold range of sensitivity, from chickens as one of the most sensitive species to mallard ducks, which again, 1,000 times less sensitive. Through years of investigation, it was ultimately found that the source of this difference in sensitivity could be linked to just a tiny difference in the genome, basically 2 amino acids in the structure of the aryl hydrocarbon receptor that binds the dioxin, or the dioxin mediating its effects. Chickens have an isoleucine serine genotype at these 2 key positions, while moderately sensitive species have an isoleucine alanine genotype, and insensitive species have valine alanine. So again, just 2 minor amino acid substitutions leading to a 1,000-fold difference in sensitivity. But with this knowledge, we can now look at the genotype of various birds and even other organisms for which these sensitivity factors have been identified, and we can start to predict where they'll lie along that scale of sensitivity. So in general, all this accumulated knowledge and these approaches that we're taking are intended to make sure that most of the chemicals we interact with in our modern everyday life is down on this low end of the curve, where we can continue to live a happy, healthy life in the presence of these chemicals that we use every day, and avoid becoming the walking dead. So with that, I thank you all for listening to this presentation tonight and for attending our special scientifique. And I wish you all well. May your repair processes outpace the damage that environmental chemicals around you are causing. And just a reminder with Halloween coming up to keep an eye on those bowls of candy with your pets around, because theobromine in chocolate, even though humans metabolize this quite well, can be not metabolized quite so well in our pets and can make them very, very sick. So thanks for your attention and happy Halloween, everyone. Be happy to engage in some discussion. Thank you.
All right. Thank you very much, Dan. That was awesome. So for the folks that are here, I think the best way to do this is, let's just have it operate like we would any other discussion. So go ahead and unmute yourself and ask any questions that you'd like to ask.
So can I ask a question?
Please do.
Joe TG, what's that costume? It's looking good.
Well, I know I had at least a couple of questions, so I wanted to give some other folks a chance to get in. I guess one of the things that's really interesting to me is thinking about like where you talked about the future and where we're headed with a lot of this stuff. And so you mentioned different assays and how basically we've moved from sort of whole animal testing, or we're trying to move from whole animal testing to sort of these cell-based assays. Can you talk a little bit about like how those cell-based assays are developed, and how like the cells that are in, like, and maybe that's too broad of a question, but like how the cells that are in those assays are like created? Are they harvested from somewhere? And yeah, I'm sure that's its own Cafe Scientifique version 4, but if you could address that, that would be great.
Sure, there's actually a wide variety of cells and cell types that are used. They range in different types in terms of their tissue of origin. So there's cell types derived from, say, breast tissue, adrenal gland tissues, liver tissue. Many of them are immortalized cell lines, so they're basically tumor cells that can be grown perpetually in culture. Some of them are genetically modified or recombinant cell lines that have had reporter genes inserted into them. So for example, using things like firefly luciferin or green fluorescence protein that are now under control of different genetic elements to basically respond to certain signals and binding to certain receptors or activation of certain transcription factors. There are some assays that actually use primary human cells, so collected from biopsies and so on, and kept in culture and used for certain kinds of tests. There's a whole variety of different cell types that are derived from various rodent species, rats, mice, in some cases bovine cell lines. So it's not just one type, there's a whole diversity used for different purposes and different assays. And I guess that's one of the hallmarks, if you will, of that approach to testing, is that it's not just one assay in one animal, but it's a whole battery of assays that encompass sort of these toxicological pathways that can lead to effects, trying to provide a reasonably comprehensive coverage of that.
Thank you for addressing that. Yeah, it sounds like, yeah, there's a lot to that.
Hey, Dan, can you hear me?
Yep, I can hear you.
I have a morbid curiosity question. Why were the cattle given diclofenac?
Basically to treat disease. So again, diclofenac's anti-inflammatory, so used to help treat disease or discomfort in these cattle. But I guess cultural practice is there when the cattle die, just to leave them in the field for the vultures to consume. So basically used as a veterinary treatment.
Mm-hmm. Thank you.
Yep.
Dan, this is Chris. We've met before, but I'm his fiance. So, question for you is about, as you guys are doing more of this computational modeling for the toxic effects in compounds as time goes forward with less animal testing, do you anticipate your start periodic animal tests to validate your computation to ensure that sort of the compound assumptions that are into this thing pan out properly? We're not missing some as unforeseen, variable interaction.
Yeah, you're breaking up a little bit, but I'll see if I heard your question correctly. So with the greater use of this kind of cell-based testing and the use of computational approaches to predict toxicity rather than directly measuring toxic effects in model animals, is there, as we reduce the amount of testing, will there still be some animal testing done to sort of verify that we're not missing important things, not missing critical events or pathways? And I do think that for the foreseeable future, at least for very high risk situations or very high profile chemicals with high economic value and so on, that these predictive approaches will be used to help focus testing resources, reduce the overall amount of testing with animals that's required, but that's still some very focused, hypothesis-driven, very targeted testing might be needed to have confidence in the decisions that are being made. As far as preventing or accounting for the potential unanticipated effects, again, we do that to the extent that's possible, but we're always limited in our decision making, whether it be in toxicology, chemical safety, environmental health, or any other area, engineering, for example. We're limited based on our understanding and our available knowledge. And there, are things that are going to get through the filters, so to speak. There's just aspects of biology that we still don't understand and can't necessarily predict. Even with our animal testing, there are things that get through, things that we miss. We can't test every species out there. So we, had we tested oriental white-backed vultures in the laboratory with a toxicity test, we could have characterized what their toxicity or that toxicity of diclofenac to that species, but we simply can't test everything and anticipate all of those scenarios. And so, it's inevitable that there will be some of these idiosyncratic toxicities, and really the goal is to catch most things most of the time and keep those events of mass poisonings or really adverse consequences to a minimum.
Okay. By the same token, expect that with more and more work, you get able to say that for a given signaling play, if you have a conserved gene sequence involving whatever, metabolic path that you get across species within a certain confidence, if where the uncertainty species, family, genus stuff is going to require or dedicated sort of analysis work.
Okay, that question was breaking up pretty badly again, so I'm not quite sure I caught it all. It sounded like you were asking somewhat about conservation of species, so across phylogenetically similar species, if we feel more confident in extrapolating to species that are maybe more related, or for which, if we know something about the pathway through which toxicity is occurring, and we know that pathway to be fairly well conserved or maybe not conserved among those organisms, that would then give us some bounds of confidence around our estimation of whether those species would be sensitive or not. I think that's what you were getting at. And if so, I think that's exactly what we're trying to do with these predictive approaches, where we do understand what the mechanisms are by which a chemical is acting. We do try to look across species and use information from their genome, from understanding of their physiology, and even from the similarities in their ecology and the exposure pathways and the similarity of their life cycles and how they might encounter chemicals, to understand something about their potential to have some adverse effects or encounter those chemicals at a concentration that will cause problems. So we certainly try to take that kind of information into account.
That's really cool. Well, I appreciate it. Thank you.
Yeah.
Dan, is there like a central sharing of knowledge, like a toxicology Wikipedia site that is the dominant one most everybody gets to go to and not have to reinvent the wheel? And then secondly, if you get around to it, in the world of toxicology, is there a way to identify where the primary sources of money to pay for study comes from?
Okay, so I'll take the first of those first. So in terms of whether there's a one-stop shop, if you will, for toxicology information, not universally, but there are various shops or online sources. So when I talked about cataloging these adverse outcome pathways, that is actually a one-stop, globally harmonized one source where people are assembling all this toxicity information using that particular framework. So trying to capture information from all types of biology literature, toxicology literature, and assemble it in a way that describes these adverse outcome pathways. That's being done in a systematic and coordinated effort sort of globally, and we're making an effort to put that all in the same place and publicly accessible. Anyone from the public can go to that site, which is aopwiki.org, and you can view all the information that's there. It uses a crowdsourced model very similar to Wikipedia, but scientists contribute their knowledge, their understanding, evidence, and assemble that on that site to help with the interpretation of these new sources of toxicology data. There are other types and sources of toxicity data. So for example, EPA curates what's called the Ecotox knowledge base, which captures a whole variety of published literature on the toxicology or the toxicity of certain chemicals to fish and wildlife, aggregates that in a database, basically reads through the papers, codes that information in a machine readable format, and that's accessible for regulatory agencies, for state, local agencies, industry, anyone around the world essentially to access and utilize that information. The high throughput testing information that's produced, all those data are made publicly accessible through EPA's CompTox Chemicals Dashboard. That's a fully open public source that's used worldwide. There are another number of other databases like that that are developed and managed by different, different countries or institutions. Like Health Canada, for example, has some of these types of databases, as does the European Union. But there are sort of not one single one-stop shop for all that information, but there are definitely globally sort of aggregated sources to find certain types of information. And then your second question, if you could just remind me again.
Is there a primary place where the money to do the study comes from, or is it a matter of just anybody all over the place?
It varies quite a bit. A lot of the new high throughput methods, that kind of characterization of toxicity is really being done mostly through large federal programs that can basically put forth large contracts to have these large batteries of assays done. A lot of the research that leads to fundamental understanding of these toxicity pathways or uncovers things like the cause of this idiosyncratic toxicity in vultures or whatever, a lot of that's done by university researchers. They do a lot of the fundamental research on different pathways, develop different types of assays that are then moved into kind of a more scaled up testing mode by some of these federal programs, and utilized in industry as well. Industry does a lot of their own safety testing. So they certainly fund studies within their own organizations, generally to test the safety of new products, new chemicals that they're developing. But a lot of that information doesn't get shared outside of those industries because it's confidential business information. It may be shared with regulatory agencies, but those regulatory agencies aren't allowed to share that data outside. And so, there's a lot of toxicity information that is proprietary. So the sources of funding, again, come from taxpayers, come from various private institutes, come from private industry. So there's a whole variety of sources, and most places where these results are published would disclose what the source of funding was, but there's not, to my knowledge, a simple website or something to go to just kind of get a listing of who funded all the studies and that sort of thing.
Hey Dan, Clayton here. I have a question. So you work since you work in like the regional EPA office as a toxicologist, is the research that you do with the EPA, is it like regionally based, or do they kind of like have like a main hub where they say, okay, all of you toxicologists study this thing, all of you study this thing? So is it like regionally based, or is it just kind of determined by someone higher up?
It's not regionally based, but I say that in the context of, so the laboratory here in Duluth is part of the Office of Research and Development in EPA. And the Office of Research and Development is basically a federal organization. It's national in scope. And so our laboratory focuses on particular issues. So the laboratory here in Duluth is basically focused heavily on freshwater ecotoxicology and freshwater ecology. That's sort of the expertise of our staff. That sort of aligns with our geographic location right here on the Great Lakes. But the issues we deal with are national and international. So for example, the laboratory here in Duluth developed a lot of the test methods, aquatic toxicity test methods that are used nationally and indeed globally for the evaluation of the safety of chemicals to the aquatic environment. And so we're not strictly regional in our focus in that respect. EPA also has what's called the regional offices. There are 10 regions of EPA around the country. We happen to be in Region 5, who's headquartered out of Chicago. That regional office is actually part of EPA's regulatory arm. So we in the Office of Research and Development do research to support EPA's regulatory mission, but the regional offices located around the country have a set of states where they work with those states to administer the federal environmental laws. So whether that be the Clean Water Act, Clean Air Act, Federal Insecticide, Fungicide, and Rodenticide Act, and so on. It's the regional offices working with the national programs in Washington, D.C., then administering those on the regional level at the level of states and so on. And so the regional offices of EPA located around the country do have a regional focus, and they're sort of the boots on the ground doing the things like permitting, a lot of community outreach, a lot of kind of the administration of the laws with the various regulated parties within those states, while the Office of Research and Development basically may have specialized expertise at labs at different parts of the country, but those labs work on national and basically issues of national and international scale according to their expertise.
Cool, thank you.
Yep.
I had a question. So I tuned in a couple minutes late. I might have missed some of this, but a few different times in the presentation, you showed kind of a sigmoid curve, which I'm assuming is like a higher concentration of the toxin in some organism, and then like the magnitude of the impact. My question is that sort of sigmoid curve that like scoops up and then levels out at the top, is that pretty universal across every toxin that we run into as a species? And also sort of what are the parameters you look at when you're building a curve like that for some unknown toxin before you would do any testing to like put some points on that curve?
Okay, so to answer the first question, that sigmoidal shape, particularly on a log concentration scale, is typically the shape that we get for most toxic effects in organisms. And that especially holds true if you get to what we would call very apical endpoints, but basically close to directly observed toxicity effects like death or reproductive failure or pathologies, incidents of pathology. They tend to generally follow that, at very low doses, almost no effect, just a background level. Then you get to some range of concentrations where there's a pretty sort of steady increase with increasing concentration, and then you reach some maximum. And obviously, if you're looking at lethality, for example, you reach a point where you're just killing all the animals, so you can't go any higher. But generally, there is some maximum that you reach. As we get down to looking at cellular responses, biochemical responses, gene expression, those concentration response relationships can be a lot more varied because of the dynamics of feedback and the more rapid control and more rapid responsiveness of many of those endpoints. And so you tend to get much more complex functions at those lower levels of biological organization, or less, less derived or less integrated sort of effects. So let me see if I can remember the second part. Sorry, I get talking, then I forget. So could you remind me?
The second part was just, so let's say you are observing some new toxin or you're studying some new toxin. When you're, I'm assuming that you build some sort of version of that curve in order to determine, in what concentrations are we okay and what ones are we not. what are like the parameters you sort of look at when you're building a curve like that?
Yeah, it's still almost always based on data. If we don't have toxicity data for a chemical, we would look at its structure and look at data that has been collected for chemicals with a similar chemical structure and do what we call read across, and assume that chemicals with similar structures will often have similar properties in terms of their toxicity. And that would be the beginning of sort of a hypothesis about what the likely toxicity of that chemical is, what the shape of its concentration response curve would be. But ultimately, we generally have to do some testing to get a sense of whether it truly follows that shape or not. And part of what goes into that is when we predict that toxicity, how does that concentration compare, the concentration at which we predict some toxicity might start to occur, how does that compare to the concentrations that we're actually likely to encounter, either from an environmental exposure, if you're talking about human health, what you might encounter in your food or in an occupational exposure. And if that margin is large, where it's really unlikely that you would ever encounter enough of that substance to get even close to that toxic range, we may call that very rough estimate good enough. But if we're approaching that level where that concentration or the concentrations we're exposed to can actually exceed those concentrations where we expect to start seeing effects, then we would typically focus or target that to collect more information. And that's generally done through additional testing, whether it be through some of these high throughput methods, or when we need to be more certain, then doing some focused animal studies.
Cool, thank you.
Sure.
All right, I'm going to jump in here real quick. We're getting pretty close to the end of the hour, and we had a couple of questions come up in the chat. So I think we'll ask those final two questions in the chat, and then we'll let Dan off the hook. So Dan, the questions in the chat, one of them is from Abigail, and first of all, she said sorry for the poor connection before, but so this is more of a personal question. What toxin do you find most fascinating? And is it man-made or naturally occurring? So are you into any particular toxin?
Oh boy, that's a good question. Most fascinating. I don't know that. I've never really focused a lot on one particular chemical, and actually for me the most fascinating thing has been just learning about the biology and how the biology responds to these different toxicants. I guess if I had to answer that, one of the chemicals I've worked with probably the most is a chemical called fadrazole, which inhibits an enzyme called aromatase, which is key to the synthesis of the reproductive hormone estrogen. And we've done a lot of studies over the years on the effects of this chemical and the inhibition of aromatase. And we've really found, what to me are really fascinating behaviors in terms of what the initial impacts of that chemical are, how organisms then adapt to the effects of those chemicals through feedback processes within their neuroendocrine system, how they change the expression of genes, how they change the production of other steroids to compensate for and account for that effect. And then if you continue those exposures, how that then overwhelms those adaptive responses and starts to impair physiological processes that support reproduction and so on. And just kind of the fascinating biology that goes into that response to stressor, and then the struggle, if you will, of the animal to maintain its normal capabilities in the face of a stressor. And I guess that's the part, more so than any specific chemical, that's the part that I guess has always fascinated me, just kind of this understanding of the intricacies of biology and how well equipped animals are to deal with a variety of different kinds of stressors and insults, but yet how vulnerable we can be when you come in contact with the wrong thing at the wrong dose. Some of these things can be exquisitely toxic and, lights out. So it's just really fascinating to study, I think.
Thank you. Yeah, that's very fascinating. Thank you.
Mm-hmm.
All right, so last question here is from Dave. And Dave's question is, are there substances that are only toxic when combined but harmless by themselves?
Yes, absolutely. And that can be combined in multiple senses. There are chemicals that may combine through a reaction to produce a toxic product. So there's like some household chemicals, I forget exactly, it's like bleach and ammonia or something like that. I might be wrong about those two exact agents, but there's something like that. There are two very common household cleaners that independently are fine, but you put them together and you get a very potent toxic product. There are also chemicals that in the body of an organism can combine to cause what we'd call a synergistic effect. In some cases, for example, you might have a chemical that inhibits a metabolizing enzyme that's used to detoxify another chemical. If you inhibit that metabolism, you can now no longer get rid of that toxic chemical quick enough to prevent its effects. And so the combination of those two can lead to toxicity. And I think, again, if we look at the labels on various pharmaceuticals that we ingest, for example, there are often warnings on the label not to combine them with alcohol or combine them with certain other drugs, because of those kind of drug interactions that can occur. The same types of things can occur with contaminants or pollutants that we encounter in the environment. Sometimes they can combine in ways that make an organism more susceptible to the effects than they would be to either substance alone.
Cool.
And so I see Elizabeth B. confirmed that I was correct about ammonia and bleach
Cool.
forming chloramine gas. So thanks for backing me up on that one.
All right, folks. Well, we're at the top of the hour here. So first of all, yeah, let's certainly give Dan a round of applause here, either, quietly in our homes or in our hearts or by the emojis or whatever we want to do. Dan, thank you so much for coming out and we're staying home and doing this for us. Really appreciate it. One thing that I want to mention quickly, so I've got a couple of things I'll mention quickly in wrapping up. One of them is that Dan was talking about sort of some of the research that's happening at our lab here in Duluth in the Twin Ports. And he mentioned, the development of some of the aquatic toxicity standards and some of those tests that are used around the country and around the world. And he mentioned briefly in his talk about adverse outcome pathways as a way to organize sort of this cascading set of events. And it's important to point out that Dan is one of the, and other folks here at the EPA lab are some of the founders of that concept. And it's kind of hard to understate the impact that organizational scheme has had on sort of the world of ecotoxicology and starting to come into human health, in the sense that just having an organizational way to start to store all of this information. And you could hear as Dan was talking about the last chemical he was talking about, which I forget the name, which is, you've got some exposure and it causes this effect, which causes this effect, which causes this effect. And keeping away, having a method to keep track of these things in sort of a standard format has really been groundbreaking in toxicology and ecotoxicology. So, and Dan's one of the founders of that. And so we're really lucky to have Dan here talking with us and as a part of our community. And so I wanted to point that out. And yeah, so so thank you again, Dan. And moving on from there, sort of the last thing I want to say is, again, thanks to everyone else for joining in. Thank you for asking some really great questions. We're going to do this again next month, hopefully with more lights on from the speakers here, certainly less creepy. We've got a talk coming up that's talking about the clash of sort of human wildlife impacts within the wetlands here. And this is, and that will be actually the middle part of next month. And so if you're interested in that talk, go visit our website or go visit our Facebook page. Dan has agreed to allow us to post this video on YouTube. So if you're interested and want to come back to some of the topics that we talked about tonight, we'll get that up later on. And feel free to reach out to myself or any of the other Cafe Sci organizers if you want to follow up on anything. I'll post some of the resources. You know, Glenn was asking a question about what are the resources, some of the databases we have for this information. I'll post links to that for anybody who's interested. I'll put those up on the Facebook page and maybe in the YouTube comments. And I think with that, we'll sign off and everyone have a safe and enjoyable Halloween. And again, thanks for coming out tonight.
Yep. Thanks everybody for tuning in, and sorry we couldn't all be together in the room to enjoy some beers together afterward and talk some more. But thanks for coming out.
Good night, everyone.
Thanks,
Right.
Dan.
Thank you.
Thanks, Dan.